So, good afternoon, and thank you for the opportunity. Now, we are going to present our project about the Angelman Syndrome. But before we are discuss about the Angelman Syndrome, first we are going to talk about the chromosome, the genes, DNA, and RNA. So this is our property that we've already made, and this is the chromosome. And inside the chromosome contains DNA and RNA. So DNA is double-stranded molecule, and DNA contains information about genetics in human bodies. And as we can see from the pictures over here, DNA is a double-stranded, and it has four main features, which is adenine, guanine, cytosine, and thymine. The differences between DNA and RNA is DNA is double-stranded and RNA is single-stranded. Also, there are differences in the features, which is the uracil in RNA and thymine in DNA. And DNA doesn't have the hydroxy groups, so it is deoxyribose, and RNA has it, and that's why RNA it's also known as ribose groups. DNA and RNA both have main role in the synthesis protein process, but as we all know that synthesis protein process is not always well intertwined, and there are many genetic disease occurs to the uneven process of synthesis protein, and one of those is the Angelman Syndrome.
So the Angelman Syndrome is a genetic condition that affects the nervous system and causes developmental delays, intellectual disabilities, speech disorders, and problems with movement and body balance. Risk factors are conditions that can increase the risk of getting this Angelman syndrome are both parents' genetics and family history of this disease. The complications that this syndrome can cause are difficulty eating, hyperactivity, sleep disturbance, the curving of the spine, are scoliosis, and obesity. Angelman syndrome is a genetic disorder that is not often inherited by parents, but does not rule out the possibility of still being inherited by parents. Angelman syndrome is caused because a gene called UBE3A that was found in chromosome 15 is completely unformed or formed incompletely, so it cannot function properly. The UBE3A gene in humans functions to send signals to the brain during the period of human growth and development. Please note that in humans, the UBE3A gene that actively sends signals to the brain comes from the mother's gene, while the UBE3A gene that comes from the father is inactive.
Angelman syndrome is caused by a disorder that occurs in its genotype. There are four main causes of Angelman syndrome, especially in the UBE3A gene. First, the UBE3A gene is deletion or deletion. There are 70% of the total cases of Angelman syndrome in the US caused by deletion. So this is the most common cause of Angelman syndrome. This deletion occurs when a copy of the UBE3A gene derived from the mother is absent or erased in the gene of copy result. The second cause is a mutation of the UBE3A gene. There are 11% of the total cases of Angelman syndrome in the US caused by mutations in the UBE3A gene. This mutation process occurs randomly and is triggered by a provoking factor. This mutation in the UBE3A gene inhibits the gene expression process of that gene and interferes with its function.
So next, the third cause is the disability of the imprinting center inherited by the mother in chromosome 15. As we all know, the imprinting center is a place where the chromosome regulates whether the genes in the chromosome are active or not. So in this case, even though the UBE3A genes is well copied from the mother, it may not be able to send signals because the imprinting center of the chromosome is not working properly. And the fourth case is the parental uniparental disomy. This is a condition where the UBE3A genes only come from the father. And while the UBE3A genes that can send signal to the brain only come from the mother, even though the UBE3A genes that come from the father is not going to be able to send signal to the brain and turn off. And for those that doesn't get the gene from the mother, the brain will not get information needed from the UBE3A gene.
In summary, Angelman syndrome is classified as a genetic disease because Angelman syndrome is caused by abnormalities in genes. The abnormality affects the process of gene expression. For example, the deletion process that eliminates the UBE3A gene so that the gene is constrained in sending the final result to the brain, or mutations that will later send incorrect protein information to the brain, as well as defects in the imprinting center, and parental uniparental disomy, which results in the gene being inactive and malfunctioning so that the brain cannot accept and continue the initiation process.
So that's all for our presentation. Thank you for listening. Is there any question?
Kalau ada pertanyaan, ntar boleh bahasa Indonesia.
Oh ya, oke. I think it was a great presentation from you, all of you. And actually, this is a very rare disease because I myself, as a clinical neurologist, never found this kind of patient like this. I don't know in the pediatric department because at our department we have also a division called neuropediatric department, which is should treat the patient like this. But during my clinical experience, I never found the patient like this. So this is I think this is one of the rare genetical disease in our clinical setting at our hospital, in Professor Kando Hospital. So this is for me, this is not about your school task for you, but also this is a refresher, refreshment for me to again read about this very basic genetic subject. So I think it was again all of you had made a great presentation, but please keep in mind that in our clinical practice there are many genetic abnormalities like this, but Angelman syndrome is a very rare disease. So maybe one of maybe I don't know who among you that is interested in medical study, but maybe you also will have a very few patients like this in the future, because the diagnostic procedure for the disease like this is very demanding, highly demanding procedure, and we are especially in our faculty of medicine in Samratulangi University, or also in Professor Dr. Erdekando Hospital as a national referral hospital, we also still lack of diagnostic tools for the disease like this. But I hope maybe one day you will become a doctor, and then you will be a pediatric or a neuropediatric. Someday maybe you will find a great way, a simple way, but still valid to diagnostic the patient like this. So I think I will not ask any question from you. I'm impressed with your presentation, and I don't know who chose this this topic for you, but it's a great topic to be deeper more as a refreshment, as I said before, about the genetic disease like this. Okay, thank you.
For the feedback, maybe in a scale from 1 to 10, how great our presentation was, especially about the information that contains in our presentation.
I think each of you have a different style in make a presentation, but we are also very limited situation to be a good presenter in this situation. So I think it's good, your intonation, your body language, and also you try to make eye contact, but I noticed that several or maybe most of you still have to read the material. So I think in the future it is better if you already maybe memorize the material, and also make a self practice before make a presentation. But as I said before, yes, we are very unconducive atmosphere to be to make a good presentation, so it's okay. For me, it's good, quite good. 1 to 10, 8. Is that okay? 8 each of you, I think 8. 8.5 for Daniel, of course. Okay, thank you.
Okay, well, that's all from us, and once again, thank you for the opportunity. Thank you.